All right, welcome back, everyone. So, in the past, at the beginning of the first chapter, we spoke a lot about, and generally, what biology is, and it's literally the study of life in this world and basically on Earth, and we then had to define what life is. Okay, so if we want to study life, we need to define what life is, so we can study that. So now, I want to talk about more. How do we study this? Okay. So, the nature of science, one thing about science, it really aims to understand the natural world through observation and reasoning. It all starts there. We observe something, we are curious people. Literally, we are very curious beings. We love to look at things around us, to observe, and always to think on reason why is that particular thing that I'm just watching is happening. So, for example, why is a frog, why when it jumps, it is able to get such a high, literally height, when it literally jumps, like how can it get that like that up? And of course, you start realizing that okay, its legs are extremely bendable. They're putting so much force on of this table, so the table has to be given some force to it to go up, right? And then that's the Newton's third law came out to be. So we are always there trying to observe things and trying to reason of it. But the thing about also one thing that I want you to also know is that science is descriptive. We try to describe everything we see, even though it's not actually the right thing, but we try to describe it in a way that we can able to understand. I'll give you an example. The classification of all life on Earth. We have this project that we want to literally classify every single living being into some sort of group by the physicality, by how they look, by the genomes, by certain things. We want that. But does it mean that we are called Homo sapiens, that that's the absolute truth? No, that's not the absolute truth. But that's something that we created, that we described. Hence, it's descriptive. We need that. We need this kind of descriptive things because this is how we understand. So that's why science is purely descriptive. We need to describe something. Another example that is brilliant is the human genome sequencing that I was talking about previously. The genome sequence is, how can I put it, massive amount of DNA. When we basically look at the results, it's purely descriptive because we see a bunch of letters A, C, T, and G's, literally all stuck together. But is that the real thing that you're actually seeing? So if I go right now, open up a cell, take the DNA, am I really gonna see A, T's, and G's? No. I'm gonna see the molecular uh, kind of structures, but not really the letters. So that's again a descriptive thing. We've described it in a sense that this part of the genome has this sequence that is ATTTGCC whatever. We're describing it. So that's one thing that yes, science starts always with by observing something and reasoning it, and we always try to describe it. We're always there trying to describe natural phenomena, natural things that are happening around us. So that's really, let's say, the essence, the nature of science per se. So when it comes to science, scientists we like to use two different types of, kind of ways of reasoning. We use deductive or inductive reasoning. Deductive reasoning, it literally means you take a big, a very general principle, and you make specific predictions. So you start by something so specific, you know already a general principle that is very general, and you use that then to do a specific prediction. Like here, all dolphins are mammals. You know, all mammals have kidneys. Therefore, all dolphins have kidneys. We start off by the biggest or the most general principle that mammals have kidneys. So you're going for something so general, all mammals. That's a very general topic. It's not specific. So okay, and then you see dolphins are mammals. By deductive thinking, by using something so general like mammals have kidneys, and then we're like, okay, dolphins are mammals though. So technically, dolphins have kidneys. So you're basically literally reasoning that dolphins have kidneys, something that is so specific from something that's so general. Now you can do the opposite. This is inductive reasoning, where you're using something so specific to give you a general conclusion. So it's literally the opposite way of doing it. So, an example. Some genes in fruit fly, for example, Drosophila melanogaster, can cause dramatic changes in development. These genes have been identified in all multicellular animals. Hallbox genes control developmental fate in animals. So let's break that down. Seems a bit complicated, but it's not. So literally, what they've seen is that in these Drosophila, uh, basically fruit, they have certain genes which they've called the home box genes. These home box genes, they saw that if they played around with it, they could play a lot with the development in a way that if we let's imagine they mutate one of those genes, suddenly one of the legs is literally growing on the eye of that fruit fly. So they're like, oh damn, these genes are really needed for the development of this fruit fly that I cannot mess this up. Then they went on to see the sequences of other animals, and they're like, ah, but other animals have these home box genes that look so similar. Okay, so from the Drosophila's observation that is something so specific, I'm making a bigger claim that, you know what, these genes that I saw in the Drosophila, that is such a small little thing, suddenly all these big animals have basically these genes, these home box genes that control their developmental fate. So this is again how you use something so small and then you turn it into something so big. Give you an example. Another one is who used to use this a lot. This inductive reasoning is Sir Isaac Newton. Newton used to basically, of course, like there's the whole thing of him sitting underneath an apple tree and the apple fell in his head. Well, he used inductive reasoning there technically, if that was ever true that ever an apple fell to his head and he came up with an idea. But let's imagine that it is. The apple hit his head, and then he started to think, but why did it fall? There has to be some force that made it go down. Well, that's gravity. So he started to say, like, okay, from this small apple, it fell down. All the objects is governed by gravity, or basically there's some gravity force pulling them. So something so small like the apple to everything. So you always have to look at the kind of direction of how the reasoning is going. So if you start from something so big, like for example here, if let's imagine all dolphins are mammals, all mammals have kidneys, therefore all dolphins have kidneys. Okay, so you look at it as like, what is the biggest thing here? Well, mammals. Mammals is a bigger group than just dolphins. So they're like, okay, we started off by a general principle and we went then to a small principle. Okay, deductive. If it's the opposite, we start off by Drosophila, then to do all animals, to all multicellular animals. No, we use inductive because we use something so specific to make a general conclusion. So in the test, I will give you examples and I'll be asking you like, hey, is this deductive or is this inductive? Always look at the rate. So what principle are you using to make a reasoning? If it's something really big compared to the other, then it's deductive. If it's something so small, then it's inductive. So please look into that. One thing about science, is that okay, we observe something and we want to reason something. The way we reason is we create a hypothesis. What we do is we create a hypothesis which is an educated guess. It's a guess in where we're trying to predict why this thing is really happening. And then we need to basically experiment it. We always need to experiment this. So we start with an observation. We create an observation. We create an observation, and then we say a hypothesis like, okay, this frog is able to jump because there is a force in this let's say table that is literally helping it to go up. That's my hypothesis. And I'm predicting that, of course, if I don't know, if I remove this table, it's not going to be able to do so. So I experiment it. So I do that. I literally basically remove this frog and put it in air. Can basically it jump from air? I experiment and I conclude. That is literally how the scientific process is. Of course, like it's not just as simple as that, but it can be as well. And what I'm simply trying to say is that yes, in this case, you can do basically one simple hypothesis, do a prediction, experiment it, and get a conclusion. But in many big experiments, and if you're trying to, let's say, prove something really big, one kind of experimentation is not enough. You need to do multiple experiments. You need to have multiple hypotheses and multiple experiments. And the best conclusion, or let's say a conclusion that you come up with and you say, and you want to really prove it to the scientific community, the best thing to do is to have multiple experiments that say that is actually showing that, and other experiments that are showing that other hypotheses are wrong. Because what you're trying to say is that, you know what, all different other things are not true. And I'll give you here a really nice example. Let's imagine you actually walk in to a class. You know, you're walking into the class and to your classroom, and suddenly you go into the classroom and it's pitch dark. You're not able to see anything. So your first observation is like, okay, it's dark. And you start to think, but why is it dark? Okay, I come up with three hypotheses. My first hypothesis is simply because, you know what? Someone didn't turn on the light switch. Okay, hypothesis number one. Hypothesis number two is that maybe the light bulb is not working. Maybe it's done, it just got burned. We need to change it. That's your second hypothesis. Your third hypothesis is that, you know what? Maybe I went blind. Something went wrong and I'm blind. So you start to predict. It's like, okay, I need to do some sort of prediction about this. Like, okay, if I do something thing, I'll get an outcome. So I'm also going to say I predict if I turn on the switch, it should turn on. If it's the whole problem of the switch. So I experimented. I go in, I turn on the switch, and yeah, voila, we have lights. We're like, okay. The conclusion is that yes, it was because of that. Of course, if you want to make it much more stronger and make claim that make it, let's say, bulletproof, you actually reach, first of all, is that when you turn on the light, you start to see, okay, that experimentation that I'm able to see, that it, so here you're basically, your hypothesis is that if light comes up, I predict that I will actually start to see. So you experiment it, you turn the light on, and you're like, ah, I'm able to see. So no, I am not actually blind. So you remove that hypothesis that you created at the beginning. The second one is that if I turn on the light bulb should be on, and it works. And yeah, you remove the other one. So the three hypotheses that you had, the experiments that you did, all showed that the other two hypotheses, the one about you being blind and the light bulb not working, uh, are not true, and that it's purely because of the switch. So literally, that's how we do experimentations in general. So again, this is literally what I kind of explained. So you have an observation, you come up with a question, and a question in mind, and you come up with hypothesis. Multiple different type of hypotheses. So in this case, you do five. Let's say you predict something that, okay, if I do this certain thing, I will get a certain outcome. So you experiment it. And then your experiment rejects hypothesis one and four. Great, you dash them out. Then you still have the two, three, and five. You do another experiment that tells you two and three, and that gives you hypothesis five. That's literally my job. My jobs are literally doing this kind of thing. Is I literally observe something in either cancer patients or in a mouse that has cancer, and I try to hypothesize, like, if I see something that is really cool, I try to really question it and give a hypothesis, and I do experimentations until I actually come up with a proper hypothesis or confirmed hypothesis, basically. By the way, also one thing here, that this is actually a very good example because you finished, you rejected all of them, then you have the hypothesis five, you do another predictions, and you do, this person did, let's say, three different experiments to literally confirm it. Here, maybe he did an experiment that, it didn't really work that well, that he had to redo the hypothesis again, and then he came back again. So here, I'll give you an example of what of this can happen. So there used to be this kind of hypothesis that, for every one gene literally it codes for one enzyme. So there was this one gene, one enzyme hypothesis. And people were experimenting and they saw that, yeah, there's a lot of genes in our body that it literally encode for just one enzyme. And then of course he did all experiments and yeah, it came out to be like, yeah, it's true. Later on, many other scientists started to do more experiments and they started to see like, okay, but this enzyme is made of multiple proteins. It's like literally made from four proteins. And every protein is encoded by a different gene. So this one enzyme that's made of four genes is encoded by four different genes. So you're like, okay, no, it's not true. It basically, they modified the hypothesis to say that one gene, one polypeptide or one protein. So then they basically, had to basically modify the hypothesis. And later on, we started to see that, okay, some genes can actually encode for more than one peptide. So they had to then redo basically the hypothesis. Sometimes your hypothesis will be actually very backed really well with multiple experiments, but sometimes we don't fully experiment everything. Sometimes we cannot, or we don't see it that way. Then later on, when let's say new technologies arrive, new other stuff starts to arrive to experiment things even better, we start to realize that hypotheses back in the day were not 100% true. And that's what happens. Hypothesis keeps changing quite daily almost. It's very common for this to actually happen. So a definition of a hypothesis. So hypothesis is literally a possible explanation for an observation at its simplest manner. An hypothesis must be tested to determine its validity. It is often tested in many different ways and allows for predictions to be made. So, it literally it means is that it's literally an observation that you need to first test to see if it's actually valid. It's not a true fact. It needs to be tested. It has to be tested multiple ways. You need to make sure that this is a rigorous experimentation, because it's not that I literally can give you an example. They used to say, like, if you see, let's say, a bottle and you have a white liquid, and people start to say, like, oh, white, basically, because it's white, it has to be milk. That's your first experiment. You look at the color and you observe you look at the color, you note it, white, it's milk. No, there's so many different other things that can give, the color of white. So you need to test this in multiple different ways. And you need to make sure that basically you allow for predictions to be made. When you create a hypothesis, you need to make sure that this hypothesis can give you predictions that, to be able to experiment. That's what that means. And hypothesis can be changed and refined with new data, meaning that it's once let's imagine that you do 20 different experiments that all lead to this one hypothesis. It can be that in the future, new technology comes up and they start to see, like, you know what, that's not true. Sorry, that this has to be changed a bit because we found this thing now with this new technology. Things can be changed. Science is dynamic, it's very fluidy, it's not static, it keeps changing and changing. And this is what makes it exciting for everyone. Because if science was static and our hypothesis were never changed, then why the hell are we here? No, we will never get anywhere. And it gets boring. So, experiments. We use experiments to test hypotheses. And one thing that's extremely important about experiment, and this is something that me as a reviewer of papers and experiments, I look at. Experiment has to basically test only one variable at a time. So I'll give you here a nice example, okay? Let's imagine we have, let's say, a flask, and that has water. And I'm trying to experiment is like how much influence does the air have, or the temperature of the air, or how strong the air is on the basically on the water. Can it actually really change its temperature? So you try to do an experiment to do that. So the first thing you start to think is like, okay, the water is in a certain vessel. That experiment has, and every time I do the experiment, I need to have a control, something that is not changed, and that has to be the vessel. So what type of insulation the water is in, it has to be. You need to control that, and then the wind. Okay, I'm trying to see the wind. Then I can actually change the temperature of the wind, or I can change how fast it's going. I need to be sure. So I'm only testing one variable. So first of all, I only test the temperature. So what I do, let's say I do four different experiments. All of the four different experiments have the same vessel, so the same insulation. All of them have the same speed of wind, but then I change the temperatures. So that's one thing, that one variable that I'm changing at a time. But the others is literally the same, because I need to make sure that only that one variable that I changed is the one that is actually causing a difference in the temperature of the water. Because let's imagine, this is definitely not true, but let's imagine that if I keep changing the temperatures of the air, nothing really happens to the water. Okay, then I'm like, okay, it's not actually the temperature of the air. Then I change the speed, and then suddenly by seeing by the speed that, yeah, speed actually changes it. So now you're 100% sure that it's not the temperature, but it's the speed of the air. So it's extremely important. So you need to have certain controls. One important thing that you need, and actually in an experiment, is a positive and a negative control. A positive and a negative control, it literally means their name are quite telling. Positive control is a control is something that when you change, you know it's gonna give you a positive what do you call it result. So in the case of the water and basically in there, I know that if I literally put the air extremely cold, I know it's gonna basically change it. So that's a positive control. I know it. Another thing is literally is a negative control. It's the opposite. It's a control that you know that if you change, nothing's gonna happen. So that, for example, can be, let's say, um, trying out hard to think about something, but it could be basically, for example, the color of the insulation. I changed the color from white to red. No, it's not going to change, and yeah, it didn't change. This is just a test that your experiment is rigorous, meaning that the setup actually is what it's supposed to be. That when it's basically when you put something positive or something that is going to change it or give you a result, it gives it, and something that it shouldn't, it doesn't. So you're testing how rigorous that experiment is, and it's extremely super important. Give you an example here with mice. When mice have cancer, I need to have one positive control, so one therapy that I know that when I add to this mouse, the tumor is going to go down, and one basically therapy that I know that when I give it is not going to do so, and that, for example, is just saline, water with salts. So my negative control is literally the saline, the water with salts, because I know it's not going to do anything. But the positive control is, for example, chemotherapy. I know with chemotherapy, I'm going to shrink this tumor a bit. So I know that my experimental thing is should be working. So when I actually give my control, which is, let's say, my therapy, I can see what the positive should look like and what the negative should look like. It's extremely important. Predictions. So your predictions or hypotheses should make predictions, and predictions provide a way to test the validity of hypothesis. So technically, it's literally you're predicting what's going to happen from a hypothesis. So hypothesis must be rejected if the experiment produces results inconsistent with the prediction. So meaning that if basically I'm predicting, in the case, let's go back to the water, I'm predicting that, if I change the temperature of the air, I'm predicting that the water is going to be also have a change in its temperature. So I do an experiment. If the experiment suddenly basically shows that yes, if I change the the air temperature, suddenly I get to also change the temperature in the water. So that's kind of how this experiment actually proved my prediction and my hypothesis. And then basically then I start to see that okay, another prediction is that I have is that the color of the vase that has the water, if I change it, that's gonna change its temperature. I change it, oh, no, nothing, no, so it has to be rejected. So the more experiments you have that support your predictions, the more valid the hypothesis is. Like exactly what I was saying, you need to do it multiple times and show that that literally your experiments are proving your predictions, and basically the other hypotheses are being nullified as well. Let's give you an example of a hypothesis driven science. So here you have a question. What is the source of contamination that occurs in a flask of nutrient broth left exposed to the air? So there is, you have this flask, and in generally, when you have, let's say, a flask that has nutrient broth is literally the food that bacteria love. If I leave it to the air, literally leave it outside, suddenly bacteria start to grow. But then you start questioning, where's that bacteria coming from? So yes, that's your first observation. And you start thinking like, maybe the bacteria just literally spontaneously came out of nothing inside that broth. That's your first hypothesis. The second hypothesis is literally your. I think that most probably it came from the air that basically the air has a lot of bacteria, and the bacteria saw this literally food and went straight to it. Okay, so you want to basically you have this basically germ hypothesis that pre-existing microorganisms present in the air contaminate nutrient broth. That's your first hypothesis, the germ, and the spontaneous generation hypothesis. Living organisms will spontaneously generate from non-living organic molecules in the broth. So you have two hypotheses. For this hypothesis, your prediction is that when you sterilize broth, so if I take the broth, if I sterilize it, meaning that I make sure that it's clean, so I boil it and I kill everything in there and it's sterile, meaning there's nothing, there should be no contamination. And I prevent any air from entering the flask, then basically there should be no bacteria because I'm preventing the air. So if that is true, if this prediction is true, so I experiment it and then turns out to be true, then the germ hypothesis is true. If then basically here, the prediction here if for the spontaneous generation hypothesis, meaning that they just came out of nothing, is that organisms will spontaneously generate from organic molecules in broth after sterilization. Meaning that if I literally get right now a flask, I sterilize it, make sure there's nothing in there, then I close it, then I, basically make sure there's no air coming in. If this spontaneous generation hypothesis is true, there should be a spontaneous generation of a bacteria inside that broth because you don't need the air, you don't care. It's literally in there that spontaneously from the broth bacteria come out. So basically, this is literally how a hypothesis and your prediction. So you go and test it. What you do is you get this next one flask. This next one flask, what it does, it really prevents the air from going inside. It's actually they're very good to use in the lab sometimes if you don't want your broth to get actually uh, contaminated. So we sterilize it. So but we do that by boiling. We boil the broth, and now we know sure that there's nothing in there. It's clean. So we do basically we have two flasks. One that is not broken. It stays like that, so you prevent the air. The other one that you break and you let air come in. You start to see that the unbroken flask is sterile, meaning that there's no bacteria. It's like, okay, there's no bacteria. So most probably the spontaneous generation is not true. Then you see basically in the one that you break, suddenly you see air coming in, and now that you actually do have contamination. So, okay, this experimentation actually helped to give you this prediction. So this prediction is true. So then it's actually the germ hypothesis that is correct. So that's basically how you start thinking in a scientific manner. Observation, you reason, you create hypothesis, you predict something that if you do something, this is what's going to happen, and then you test it, you experiment it, and then you get basically your results, and then you conclude what hypothesis was right or not. So one thing that in scientists we like to do in general, we like to study, of course, let's imagine we're studying the human being, or let's go even a bit smaller. We're studying the heart. Sometimes the heart is extremely complex. It's very big. Really, it's made of so many different types of cells, and so many things are happening in literally milliseconds that is can be very overwhelming. So one thing that we like to do is we like to actually kind of reduce it into simpler parts. So we break down the heart into literally just one protein, and we study that protein for a bit. That's called reductionism. And actually, I did that a lot in my bachelor's. In my bachelor's, I was studying how people have hypertrophic cardiomyopathy, is when your heart muscle gets so thick that you get a heart attack. And you get to see this a lot in football players. So I was trying to study that, and my boss during that time, he didn't want to study the full heart literally in one go. He took one protein of the heart and studied just that one part. It's easier and makes things, let's say, much more simpler, and you can get a lot of information. But you have two limitations. Parts don't always behave in the same isolation. So that protein could actually some if you put it back in the heart, it behaves in a different manner because now suddenly there's other stuff there that it interacts. The complex interworking of many pathways lead to emergent properties that cannot be predicted based on working parts, as we were saying previously. This one protein that I'm talking about When you put it with other proteins, it starts to interact with other proteins to create something bigger. To let's say create a let's say an enzyme. Well, you cannot predict it by just studying it by itself. You need to put it with the others to see its emergent properties, properties that come up when this protein is literally interacting with others. So that's you have to always be careful if you're using the reductionism way of studying something. The other one is systems biology. This is more my favorite, in which basically explores the biological system by analyzing the interactions among its parts and how these interactions give rise to the function and behavior of that system. So you're really literally trying to mimic what's happening in that system. So you try to put multiple components together and you're focusing on the emergence properties. I do that a lot. What I really love to do, if I actually sometimes cannot, it's not always possible, but in some cases I can. The doctor will give me the piece of the tumor of a patient. He will also give me his blood because what I'm trying to do is because the immune system has a huge interaction with cancer and I want to test that. So I take his blood, I extract his immune system, so his white blood cells, and I put them on top of his cancer because I want to see in a system where his own cancer and his own immune system, how are they basically interacting with each other? It's extremely important, and it's really you need it kind of thing. So think about this. System biology is about putting together and integrating, while reductionism is about taking apart. So always care, remember, to fully explore emergent properties, biologists pair reductionist theory with systems biology. So basically, sometimes we do both. It's actually the best way that we might actually start with a reduction theory, that we start to look at just one protein and then we see like, okay, it has some certain properties, super cool. Let's put it in its back in its system, and then we study again its system and we see it. Once you're done, so let's imagine we're done with basically our experimentations, our hypothesis, you name it. The most important thing, and this might sound so naive and stupid, but it's the most important thing you'll ever do as a scientist when you're done creating a model. We, as human beings, we love to see things. Believe it or not, all of us have some artsy part inside of us that we are visual learners. We like to see something. If I'm here and I'm explaining to you the whole structure of a DNA by just my words, it's nothing compared to if I literally draw a whole model. If I literally or give you either draw it for you or literally build you a model like what Watson and Crick did. So here they literally built how the DNA looks like and how basically, the helical structure and all of that. That by itself is much more than words. So we do that a lot. We do that a lot, for example, for signal transduction pathways or the structure of proteins. So signal transduction pathways, if you don't know what that means, so let's take an example of insulin. When insulin, as we know, insulin is a protein that is really needed to kind of boost our own pancreatic cells, or sorry, our own cells around, sorry, to uptake glucose. So this usually happens when we eat and we have a lot of glucose in our body. It has to be taken up. So insulin does that. So insulin is able to bind to cells through a receptor. This receptor gets activated and it activates multiple series of proteins inside that cell to actually start saying to the cell, start uptaking it. That's the transduction pathway. So rather than telling you, oh, you know what, protein 1 basically binds to protein 2 basically phosphorylates protein 3, and protein 3 goes to 4. No, I don't want to say it. I just draw it for you. And that drawing is literally says 1,000 words. It's crazy how good it can be. So a model should put shows how parts fit together. Or basically how suggest experiments to test how components work together. So really, it's trying to put all the small parts there. So for example, here is a model of the plant cell. Here we can see there's the cell wall, the cell membrane. Here basically you literally have the nucleus, you have the endosomes, we have the mitochondria. With only that picture, I could see so many different structures. They're trying to put all these different structures together. So that's how important models can be in science. So one thing I also want to talk about in generally in science is scientific theory. So scientific theory can have two meanings. One, for some people, scientific theory can mean is an explanation of a natural phenomenon. For example, Gravity, the theory of gravity. You're trying to describe a natural phenomenon that's occurring. But for many people, which is, let's say, the more correct answer, it's literally a body of interconnected concepts. It's multiple concepts that are coming together and you're connecting them. This could be quantum theory. Quantum theory literally embodies a lot of different physical aspects of this universe and you put it together. So that's what a scientific theory is. It's literally multiple concepts that you connect together. But these concepts, extremely important to know, they are supported by experimental evidence and reasoning. It's not that I just randomly put a lot of concepts and I connect them together with no experimentation. That's not a scientific theory. No. And this is one thing that you need to also remember. To the general public, a theory implies that it's a lack of knowledge or an educated guess. It's not. For us scientists, a theory is literally a body of knowledge tightly coupled with scientific reasoning and experimental evidence. It's literally you're saying something that is so backed up by a lot of research. And I give you an example, Darwin's theory. Darwin's theory, people start to say that, you know what, it's actually a lie. It's not. There's so much scientific evidence that proves that it's literally a fact. So for people even call it a fact, but that theory that he came up with is not just observations. It's things that he tested, but also we basically human beings tested way into it. One last thing about science that I want to go for it is that science there's two types. There's basic and applied research. So there's basic science or applied science. Basic science is the field of science that you're trying to actually extend the boundaries of what we know. You're trying to answer questions of why, how, when, all these things. You're trying to literally, why does this protein do this? Why does the heart do this? Why do we think, or how do we think, or what time do we think? These small little things. That's what basic research is. You're trying to extend knowledge. You're trying to create knowledge by that. Applied, you use the basic research, the boundaries that what we know to create something. That's what applied is. Basic, you're just trying to answer questions. Applied, no, you're trying to use those answers to create something. So basic research, you see this mostly in academic institutions. So if you look into LAU right now, you see other people doing many different types of basic research, trying to understand how certain things work. Applied research, you see this a lot in companies, especially in pharmaceuticals. We pharmaceuticals, people go and they try to take all the knowledge that's taken from university and they try to make something, either a drug, a food additive, you name it. For me, I do both of this actually. I do these two things in my lab. I love to understand what's happening in a cancer patient. And if I started to suddenly see something that, you know what, like this immune cell, if activated correctly, it can actually kill the cancer really well and the patient survives. I developed a therapy to make that happen in other patients. So I look at basic and I try to come up with an applied all at the same time. So now I want to quickly just talk about Darwin and the theory of evolution in general. So I want to use it here basically how one scientist went on a voyage and managed to actually, just by observations, develop multiple experiments and actually get a theory out and to get it into acceptance. So basically, it all started when here Charles Darwin was at 22 years old. He took the HMS Beagle, which is literally the name of the ship, which started up basically up here in Britain. And basically, he went all down into basically the Galapagos Islands and all around basically this kind of routes. And he was going from literally place to place and observing many things. But one thing that he saw uh, basically was these kind of animals. These are the finches. He started to see that in the Galapagos and many different islands, there were a lot of these Galapagos that looked so similar, but then every island they had a different characteristics. So, example, this is the woodpecker finch. In this one island, it actually had a very long beak because and that beak was needed because it had to go into the small little holes of a branch to get its food. Then he went to another island. He saw this similar finch, but with a very different, let's say, beaky kind of or stronger beak to kind of help it to actually get these kind of food from the ground. And then he went to another Galapagos Islands, and again he saw a very similar finch, but with different complete beak, and which was like more, let's say, accustomed to get certain food from the cactus. So he started to see, it's like, okay, something is happening. We things are changing. Like it's like these animals are so similar, but they've changed. And this thing that of changing is called evolution. So literally, evolution is that living things have changed over time. But Darwin was not the first person that actually said that proposed it. Evolution was something that even before Darwin was they were talking about. Especially a lot of philosophers were discussing that things change over time. Darwin didn't really wasn't, let's say, the inventor of evolution or discoverer of evolution. No, he was more famous for giving a mechanism for change. What that change is, how that change is being driven. So how's it being caused? And he came up with natural selection. He started to see that these creatures came from most probably a common ancestor. So there was most probably a common ancestor between them, and which basically this ancestor basically flew to all these different islands, and the ones in this island, they developed their beak to actually be long to in order to get food. So there was some sort of selection that the kind of nature kind of chose the ones with the longer beak because those were able to get food and survive while the others died. So that's how basically now that species changed. While this one, when basically these ones, they basically had a certain characteristics, they survived. That's what he came up with. Another thing that also during his voyage that Darwin was having a look and really studying a lot was this essay, this mathematical essay by Thomas Malthus, which basically was titled "The Principles of Population." And what he basically said is that humans actually can grow, or like a population can grow geometrically, while food, or the food that we eat, grows only arithmetically. And I'll give you an example that means. A geometric progression is literally meaning is that it's increasing by a constant factor. So it's literally being multiplied every time by a constant factor. Arithmetic progression is that it's literally increasing by a constant difference. So it's always being added by a constant. So in this case, it's always plus two. He saw that a population can grow like this in a geometric progression. But the food, no. The foods that we basically intend to use for eating can only grow arithmetically. So he started to say, but why is that? Because if this is literally 100% true, in a sense, that if this was literally, okay, this is a mathematical model, but it's not really the case here, because throughout the years, our populations are quite similar. They don't change that much. But if this is true, we should be basically by a point have covered the whole entire earth. But that's not happening. Why? Because there's death. A lot of people die because there's not enough food. That's why we cannot basically keep reproducing into high numbers, because our food production is not at the same rate as the rate that we can reproduce. So it was literally these observations of natural of basically how this kind of finch is similar but different in these different islands because of the environment. These kind of had the whole mathematics of how a population can grow and the food grows. That he started to observe certain things. He started to see individuals in a population vary in their traits, many of which seem to be heritable. So a lot of individuals. So yeah, let's take we have a population of these kind of finches in this one island. These finches are also can be have their own different things. Let's look at us humans. We have different characteristics, and it seems that these are being passed on to the offsprings. Please bearing in mind that Darwin was not lived before the Mendelian genetics came out. He didn't know anything about genetics. This thing of genes and being things being passed on, we never knew about that. So he was already inferring it. He was just kind of saying that, in this one basically population, there's many different traits, and some of them are being passed on. The second thing that he observed, although every organism has the potential to produce more offspring, only a limited number do survive and reproduce themselves, meaning that competition is inevitable or unavoidable. As I said before, we cannot keep reproducing unchecked because our food is that will not equal the amount of people we have. Certain organisms will be able to basically be able to do so because they will be able to gather a lot of food, while others will not and die. Species generally suit their environment. They are adapted to the circumstances. So if you go back up here, the woodpecker, this finch one, has a certain beak because of the environment, while this large ground finch has a pecker made for the ground. So they are literally changing because of their environment. They are adapted to that circumstance. So he came up with, and he said, natural selection. Environment selects for the propagation of beneficial traits, hence the survival of the fittest, meaning that the environment will select for literally certain traits that actually give you a benefit and will make you survive. And these are then passed on. And this is how we change. So let's imagine we have a bunch of giraffes, and some giraffes have short necks, some have middle necks, some have very tall necks. And suddenly we start to see that the trees are growing so high that what happens is that the giraffes with the middle and the um, small necks will die. The ones with the long necks will survive and will pass it on. Hence, they basically seem to pass these basically characteristics, and it's because of the environment. The environment killed off the ones that did not survive and kept the ones that survived because they were able to reproduce and eat, and they passed this on, and then the whole species that before was all of different neck sizes has one neck size. That is the change of a species because of a natural selection. Darwin was the person that literally kind of gave an explanation of how this happens, how this change is happening, and it's really tightly regulated by the by the environment. Then basically, this was Darwin, but then way later on, literally hundreds of years later, people started to actually see the evidence for Darwin's theory, that what he was saying. They started to see fossil records. So they started to see the transitions of beings. How certain things started to change. So they started to see certain birds how they changed throughout the years because of the fossils, the Earth's age. If what Darwin is saying is true, that the environment really changes how a species looks like, or a population looks like, because of the environment, that doesn't take 10, 20 years. It takes a long, long, long time. And Darwin was always skeptical because during his time, the philosophers were saying that most probably the Earth is about 100 million years old. For Darwin, he was thinking that is too little. That is not enough. And then later on, we started to feel, I was like, no, it's much, much older than we thought. It's 4.5 billion years old. So makes sense with then basically Darwin's way of thinking. Third, mechanism of heredity. He didn't know that we were passing on characteristics. He didn't know that. We later found out Mendel's laws that we will definitely I will teach you quite a lot about. So yes, certain characteristics that suddenly were giving you an advantage in this environment were actually, being passed on. Then comparative anatomy. People started to look at different structures, so analogous structures. So the human, the cat, the bat, the porpoise, and the horse. They looked at their limbs. These are analogous structures. They look different, literally, but look at their structure and function. The inside of the bones, it's homologous. So we started to see that there is actually some homology, that we came from a common ancestor. That there is something that we basically, we came from something so similar, and then basically started to expand. So we started to see that, yeah, there is a common ancestor that basically it kept changing throughout the years, and this is like literally a bit of evidence that we came from that common ancestor. Molecular evidence. We started to look at DNA, and we started to see that certain animals looked similar to us, like the monkeys. The monkeys share almost 98% of our genome, extremely close to us. That only that 2% really changed us into the humans that we are now. So it really that evidence was very almost kind of the nail on the coffin, that this is to show that how true what Darwin was saying, that we came from one common ancestor, which was here, which we then later started to kind of differentiate because of the whole environments that we were living in. So I'm going to stop here.